acids are of primary importance when it comes to determining protein structure and function. So it's important that we know about these amino acids and we're kind of all on the same terms. When I say an amino acid name, you should know its properties and things like this. So let's go over those. Let's go over the names and abbreviations for the amino acids, their properties, and things to know about them.
There are 20 common genetically encoded proteinogenic amino acids. Don't worry, I know it's a lot of words, but basically all it says is that there are 20 of these main amino acids that our bodies use and piece together to make proteins based on genetic instructions. So 20 main amino acids, and the goal for today is to learn their structures, their names and abbreviations. So we'll talk about their 3-letter and 1-letter codes, and I'll go into some kind of memory tricks to help you remember which is which. You want to know the stereochemistry of these amino acids. So these are all going to be L amino acids, and I'll show you a little trick so we can remember how to draw them with the correct stereochemistry. We'll talk about how we can classify these amino acids as polar or nonpolar, aliphatic or aromatic, basic, acidic, or neutral, and what I like to think of as kind of normal or weird. So why? All of this is going to prepare you to see how the amino acid structure directly influences the protein structure and function. Basically, these amino acids are all going to link together through these amide bonds we call peptide bonds. And then we'll talk about how we can call this like the remainder, the residue. So when these amino acids are incorporated into a chain, we call what we used to call an amino acid, we now call it a residue. You get this long chain of amino acids, and that's going to fold up to make a protein. And the sequence of amino acids is going to be really important because they're going to, each amino acid has a unique part, a side group or a side chain or R group that's going to stick off. And because these have different properties, they're going to influence how the protein folds and therefore how it functions. So you can imagine if you have something small and flexible, you might get a flexible region of a protein, whereas you have something big and bulky, it's going to be harder to fold up. You can have various types of interactions forming. You can have things that are hydrophilic, so they like hanging out with water, those who want to be on the surface. Things that are water avoided, so hydrophobic things are going to cluster in the center, et cetera, et cetera, et cetera. And so we'll see how this protein, the amino acid sequence, which we refer to as like the primary structure of the protein, is going to directly influence the secondary structure. So that's going to be like how the backbone folds up and interacts to give you things like alpha helices and beta sheets and strands. And then how that is going to influence the further levels of the protein structure, like the tertiary structure, so how those side chains are directly interacting with other side chains or with the backbone, and then the quaternary structure, where if you have multiple chains of proteins. And so all of that is going to be stemming from these amino acids. So it's really important that we understand these amino acids and that we be able to recognize them when we see them, and know their structures. So for my 301 students, I'm going to expect you to be able to draw all of these structures from memory. For my 311 students, you will need to be able to recognize them if you see them. And for both classes, you will need to be able to know their names, their 3-letter abbreviations, and their 1-letter abbreviations. And so we'll give you some hints for remembering these. First off, let's talk about the just the basic structure of an amino acid. So you have this, you have an amino, it's named because it has an amino group and a carboxylic acid group. Except normally this carboxylic acid group is going to be in its carboxylate form, so it's been deprotonated. Basically, this carboxylic acid group, depending on which amino acid that it is located on, it's going to have a slightly different pKa, but it's going to be about 2. And when we talk about the pKa, remember that's the pH at which half of it is going to be protonated and half of it is going to be deprotonated. If you're at a pH above that, there's going to be fewer protons available, so you're going to be less of it is going to be protonated. And if you're at a lower pH, you're going to have more protons available, so more of it's going to be protonated. If we have a pKa of 2, well, this means that basically anything above 2, we're mostly going to be deprotonated. So if our bodies are about a pH of 7, we're going to have that be deprotonated. We also have that amino group, which can it can also protonate or deprotonate, and basically it's going to have a higher pKa. It's going to have a pKa closer to about 9 to 10. And this means that basically anything below that you're likely to be in the protonated state because there's going to be more protons available. So the predominant form of amino acids we're going to see is what we call this zwitterionic form. And so we call it zwitterionic when it has a positive charge and a negative charge that cancel each other out. So this is how I always want you to draw the amino acids, is in this zwitterionic form, unless I'm specifically asking you, okay, well, at what pH, what would it look like at this pH or this pH or this pH? Such as when we're drawing titration curves. But for all other times, I want you to draw it in this zwitterionic form. I never want to see you draw it in the neutral form. I know that sometimes I might accidentally draw it like this because it's kind of the convention, but this is never really going to exist, because if you're at a pH that's low enough to protonate that carboxylic acid group, that carboxylic group at that point, or into a carboxylic acid group, then that amino terminus is or the amino group is definitely going to be protonated. And if you have something, if you're at a high enough pH where you're going to have deprotonated that amino group, well, now that carboxylic acid group is definitely going to be protonated. So I don't want you to be drawing this, the kind of like non-ionic form, this where you have an NH2 or an OH. Instead, I always want you to be drawing this zwitterionic form, but recognize that you are often going to see this form used where you would see NH2 on one end and COOH on the other end. Remember that COOH is an abbreviation we often use to talk about carboxylic acid. So COOH, remember that's our carboxylic acid. Sorry, I can't spell very well. And our COO minus, that's going to be our carboxylate. So most of the time when we're talking about amino acids, we're actually not talking about an acid, we're talking about the carboxylate. But I guess you could then think of the amino group as being an acidic at that point. So when I'm showing you the amino acids, I'm commonly going to be using like figures like this, where I kind of have things kind of cartoony, and I'm not showing the stereochemistry here, but it is important to know that amino acids do have stereochemistry. They are chiral molecules, all of them except for glycine. In terms of the actual like naming for, so all the amino acids are going to be L amino acids. This L though does not refer to levorotatory, so it's not talking about rotating polarized light to the left. And D does not refer to dextrorotatory or rotating light to the right. In the case of the amino acids, it's just kind of convention that we do it based on how their similarity to D-glyceraldehyde or L-glyceraldehyde. So that's like the simplest sugar. And we say if they're laid out in the same way, if they have the same stereochemistry as glyceraldehyde, then we're going to go ahead and we're going to call it L, because that L-glyceraldehyde rotated light to the left. And so in the case of these amino acids, they get this L notation because they're structurally similar in their stereochemistry, but they don't all rotate light to the left. Some do, some don't, but that's not what the L and D refer to here. We'll also see something similar when we're talking in the context of our sugars in the naming. They're still are also going to be based off of that kind of convention comparison to glyceraldehyde. It's going to make it easier for us to kind of like compare things and not have to think about whether going to the lab and actually measure what rate of light is rotating. So we typically refer to them in this L and D notation. Now, the chiral center in these molecules is what we call the alpha carbon. And this alpha carbon is going to be the carbon that's attached to the R group, so to that unique side chain. And it's also going to be attached to on one side, it's going to have the carbonyl, on the other side, it's going to have the amine group. And then this R group is going to be sticking up and out or down and back. So if we were to draw out an amino acid, we have our H3N plus, then what we're going to have is we're going to have our alpha carbon, then we're going to have our carbonyl carbon as part of that carboxylic acid. So this right here is what we call the alpha carbon. And this is going to be our chiral center. So remember that we can use wedge to show that something is coming out at us. And so this would be our R group coming out at us. And then going back, we would have a hydrogen. And so right now what we're drawing is we are drawing basically this carbon right here. This is our alpha carbon. So it's attached to the R group, it's attached to the carboxylic acid, and it's attached to the amino group. Now, what we have here is basically, you can see an amino acid written in a couple different ways. And in this case, what we have is we kind of have this V with the alpha carbon is sticking up. So what I like to kind of think about is up and at ya. So basically, if you have this V sticking up, you're going to have the amino acid sticking out at you, the R group sticking out at you. Now, if you have one where you have the V sticking down, such as in the next amino acid in the chain, so we're always going to start from drawing the N terminus to the C terminus, and this is going to be the same when we link these up. We're going to go from N terminus to C terminus, and this is actually how they're going to be pieced together when proteins are getting made. So you're always going to be wanting to think in terms of N to C. Now, if we draw our amino acid like this, again, now this is going to be our alpha carbon, and then we have our carbonyl carbon and part of the O minus. And so here what we have is basically, we have in the R group is going to be sticking down and it's going to be sticking back. And so this is going to be our alpha carbon. This is going to be our R group. And now our hydrogen is sticking forward, but our R group is sticking back. So when we have it sticking down, so we're going to have down and back. So up and at you or down and back. Okay, so that is when you have individual amino acids. And then when they link together, they're going to form these chains. And we'll talk much more about peptide bonds and peptides. But for now, basically, the reason why it's really important to know both of these is because when you have these amino acids link up, they're kind of going to alternate going up and down. And so going up and at you or back, down and back. So what you're going to end up with is you're going to have H3N plus, then you're going to have one that goes up, and then you have your carbonyl carbon. But in this case, the OH, the second O from the carboxylic acid was going to hook up with the amino group, with the hydrogens from the amino group of another one. You're going to lose the equivalent of water. And so what you're going to end up with is now you have in the backbone, you have that nitrogen. Now you're going to have your alpha carbon sticking back. You're going to then have your carbonyl, your carboxylate group at the end. And so here we have our alpha carbon and our alpha carbon. We're going to have our R group sticking up and out. And we're going to have our R group sticking down and back. And often we kind of leave out the hydrogen, but remember that there is a hydrogen sticking back here, sticking up and out here and down and back here. But we're typically thinking in terms of the R group. So you can think of your up and at ya or down and back. And so then you just keep going throughout the whole peptide, seeing that the amino acids are kind of alternating between being up and down and being like out of the plane and into the plane. And this is going to have important implications when we talk about protein structure. So I want you to be able to recognize the stereochemistry. And then when you're drawing them, obviously you're not going to be drawing them in little cartoon form. So know how to draw and recognize them in their more conventional form, which you can get plenty of looking at in the textbook and stuff. So now let's go into some of the abbreviations. But first, let's talk about why we care about these abbreviations. And then we'll talk about kind of tips for remembering them, because some of them are very intuitive and some of them are kind of weird. So why care? Basically, instead of writing out these whole names of the amino acids, so instead of having to write out like asparagine, methionine, isoleucine, if you want to talk about every amino acid in a protein, which can have hundreds of these, well, then that's going to get very unwieldy. And so instead we need abbreviations. And so we have like 3-letter abbreviations, and then we have 1-letter abbreviations. And this is kind of a way where we can think about the amino acids, or in this case, what we call, because they've hooked up, they're no longer acids, so we call them residues, the residuals from what was linked together. So we're talking about those R groups. We're talking about typically refer to these things as like residues, but sometimes we still slip up and call them amino acids, but technically they are residues. So be able to know that we'll use that term kind of interchangeably, but technically in the context of a protein or in a peptide, what was an amino acid is now referred to as a residue. And you only have that amino group and the carboxylic acid group at the ends, except if there is one of those also present in the R group. But you can see from this simple example, or not simple example, but from this example, if we talk about like sickle cell anemia, we'll talk more about hemoglobin, but basically it's a protein that is going to carry oxygen throughout the bloodstream. And there's a mutation that causes there to be an amino acid substitution from a glutamate to a valine. And we can, instead of having to write like glutamate to valine, we can basically just write E6V to represent that the sixth amino acid residue got swapped from a glutamate, which we abbreviate E for a reason I'll get into, to a valine, so a V. And so this shorthand notation allows us to better understand this. Also, if we want to look at, say, the whole sequence of a protein, and even like look at its structure. So this is the PDB. We'll get into a lot more into the PDB. But basically what this is showing you is the structure of this hen egg white lysozyme. And right here, it's showing you the sequence. And you can see that it has the sequence in this one letter code. That's going to make it very easy to have this compact sequence that then we can look at. It's important, though, that in order for the sequence to kind of be helpful, we need to know what the sequence represents. And so if we were to see like E6V, we should be able to know, oh, that's a glutamate and a valine. So we're swapping something that's hydrophilic for something that's hydrophobic. And this might have important consequences. And indeed, by switching that to a hydrophobic thing, well, now you've introduced something that water doesn't like onto the surface of a protein, and it's going to cause that hemoglobin to clump up, leading to sickle cell anemia crises. And so we'll see examples like this, where I expect you to be able to recognize the codes so that you can recognize the implications. And in order to recognize the implications, you don't just need to know the codes, you also need to know the properties of the amino acids. So we'll talk about both the codes and their properties. In terms of the codes, the like abbreviation, the single letter codes, we'll see some of them are kind of weird, but they actually have like a reason why they were that way. We have to thank for the codes in the first place. We have Dr. Margaret Oakley Dayhoff to thank. So basically, she is kind of considered the founder of the field of bioinformatics, and she shortened these names so that the computers could deal with them more readily. And so we have her to thank for these, and so you can go learn about her more at this link that I'll provide. Let's get into some of these names, and then we'll get into the properties and what I expect you to know. Okay, in terms of the names, I'm only going to really go through the ones that are kind of weird. Some of them are very self-explanatory, like glycine, G-L-Y or G. You can see it's just the first few letters and the first letter. So basically for the ones that were more common, like if their first letter wasn't taken already by something else, then we get to give them the first letter. So you'll see for glycine, well, it's going to get a G. But then if we have something that other things with a G, well, now what are we going to do? And so we have to give them a different letter. So we'll see that, say, glutamate, it's going to have the abbreviation E for its one letter code, and glutamine, it's going to have a Q. So let's get into some of these weirdos and how we can kind of remember them. And its abbreviation is TRP, so it's not its first few letters, but it is still kind of makes sense phonetically, so tryptophan. And in terms of the W, what I like to think of to remember this is tryptophan. So think about saying tryptophan like Tweety Bird, and you'll remember that you have the W as its abbreviation. And what I also like to kind of think about is its structure kind of looks like a W if you squint a little. So that would be our tryptophan. Phenylalanine is another one that's a little weird. So it has a PHE for its abbreviation, so just its first few letters, but then its abbreviation is F. But thankfully, you can think phenylalanine, Phe, like F, so that makes sense phonetically as well. For a tyrosine, its abbreviation is TYR, and it has a Y as its one letter abbreviation. And you can think of tyrosine, it has the Y as the second letter. and the T was taken by threonine. So that's why tyrosine is going to have that Y. A couple of the other ones that are going to be weird. So basically we have asparagine and glutamine, which are going to be N and Q. So you can think of asparagine. So it has ASN as its abbreviation. So not its first few letters, but instead ASN, because ASP, that was already taken by aspartate. So ASN is going to be asparagine with in, so think N, and it's going to then have that N on the end of ASN as well. So N for asparagine, and it has this amide group at the end. So you can think it has this N here, and it's got its N in the name. The only potential complication with remembering N for that is that that glutamine also has an N at the end, and it has an N in the end of its name. So you have an N in its 3-letter code, but then its 1-letter code is going to be Q. And so basically you can think of glutamine like Q, glue, Q. So you can remember that that is going to be glutamine. You have to remember though that glutamate also has that kind of like Q sound, but this is going to be an E for its abbreviation. And there's not really an easy way to remember that other than kind of remembering that these negatively charged ones are going to be D and E. Basically for aspartate, it's like aspartate. So you can think of D. And then, well, E is going to be right next to D. And so basically you can also think about glutamate is longer than aspartate. It's got 1 more carbon. It's going to be farther in the alphabet. So it's going to be E, and aspartate is going to be D. So you can think of it as aspartate, but it's actually a tate. So ASP and D is going to be your aspartate. Sometimes you'll see this as aspartic acid, but similarly to how when we talked about our carboxylic acid group, at the end, we talked about how it was deprotonated. Typically, the same goes for aspartate. So we typically see it in its aspartate state, and you can think of it as aspartate. But remember that when you spell it, it's going to be with a T, but with its 1-letter abbreviation, it's a D. Glutamate, it's 1 more carbon, and it's 1 letter further in the alphabet in terms of its 1-letter abbreviation. So that's going to be our E. Sometimes people say like glutamic acid if you want to remember it ek, and like E that way, but that's going to be our glutamate. And sometimes it's easier for me to just remember that these are going to be D and E, that my negatively charged ones, what we call like our acidic amino acids, are going to be D and E. And if you can remember that, and then remember that glutamate is 1 longer and it's 1 further in the alphabet in terms of both its whole name and its abbreviation, you can remember that it's E. Similarly for asparagine and glutamine, these are the amide versions of these. And basically, so here we have our glutamine is going to be 1 longer than asparagine, and its letter in the alphabet. So both of them are going to be further in the alphabet in terms of its 3-letter code and its 1-letter code. And you can think of glutamine as Q, glue, Q. So that's going to be your glutamine, and then asparagine with the N. So again, there's a lot of them that have "ine" at the end, but if you just have to kind of like remember okay, well, this is one of the weirdos. What's that remembering trick for the weirdo? And then it's going to be your asparagine. And remember that one of its key features is having that amide group at the end with the nitrogen that you can then remember at the end. Okay, a couple more of the weirdos. So basically for lysine, I like to think of special K because lysines, I consider it very special, and it has this abbreviation of a K. And so basically the reason for having this K is basically that it's close enough, it's closest to L, and L was already taken by leucine. So let's give it something that was close, and so it gets this K for lysine. Arginine, A was taken by alanine, so basically arginine is going to get an R, but you can think of it as like arginine. And so you'll hear a lot of bad puns from people about what's a pirate's favorite amino acid, and they'll say arg. So basically arginine is ARG or R. The only other kind of weird ones, isoleucine, so it's going to have ILE instead of ISO as its name. It's going to be ILE. Isoleucine, you can still kind of, it makes sense phonetically and all that stuff, and it's got an I as its first letter. So that's all good. And I think that the rest of those are fairly normal. So now let's get into their actual properties. So you might think of glycine as being like the simplest because, well, it only has this hydrogen as its side chain, or we can actually think of it typically as like not even having a side chain. But in reality, this makes it really weird. And so when we think about like our most generic amino acid, we typically think about alanine, which has this methyl group as its side chain. Alanine is one of the what we call the nonpolar uncharged amino acids. So we talk about our nonpolar uncharged amino acids. I'm talking about these things in yellow. And basically, when we think about something that's nonpolar, what do we think about? We think about something that's like a hydrocarbon, right? So if you look at these nonpolar amino acids, you're going to see a lot of hydrocarbon. You're going to see these carbon and hydrogen chains, and they're either going to be aliphatic, so they're going to be like chains, or they're going to be aromatic, so they're going to have resonance stabilized rings. Tyrosine is also aromatic, but it's also polar, and so we'll get to it later. But these are going to be the nonpolar uncharged amino acids. And why this matters is these are typically going to be hydrophobic. And so we're going to see them on the hydrophobic side of our scale. Why this matters is if we think back to that hydrophobic effect, where basically if something hydrophobic gets into water, it's going to cause the water molecules to kind of cinch around it. And so if you have multiple nonpolar things, the water molecules, if you cluster those nonpolar things together, that's going to minimize the amount of water molecules you have to take out of commission, making it so that you can then free up water molecules. And so we'll see that the nonpolar amino acids are going to get clustered into the center of the protein with the hydrophilic ones, the polar ones on the outside. And this is going to be a driving force for protein folding, is going to be this hydrophobic effect, which is why I was emphasizing it so much in the previous video. And so I'm talking about our alanine, our valine, our leucine, our isoleucine, methionine, tryptophan, and phenylalanine. Okay, so cysteine, it's weird for several reasons, but one of the reasons is why. So cysteine is weird for several reasons, but one of the reasons why is in terms of just like if you were to measure hydrophobicity values, it would be classified as slightly hydrophobic, but we still classify it as a polar uncharged. And there's a couple reasons why. One is that, so we have our side chain is something like this, and this sulfur has lone pairs of electrons. And these lone pairs of electrons, what they're going to be able to do is they're going to be able to act as hydrogen bond acceptors. So they can form weak hydrogen bonds. So remember that for an acceptor, you want lone pairs on something that's electronegative. Sulfur is not that electronegative, definitely not as electronegative as our oxygen or our nitrogen, but it's still able to form some weak hydrogen bonds. And so if you were, say, to place this into water, place it on the surface of a protein, you could have a weak interaction between the proton on the hydrogen on the water and the lone pair on the sulfur. You can get these weak hydrogen bonds, so it can still be okay on the surface of a protein. It also can act as weak acid. It's actually so it's not classified as one of the acidic amino acids because it's a very weak acid, but it's not that weak. And so it's actually going to get deprotonated quite a bit in biochemistry, and we'll see that a lot. And when it's deprotonated, it makes an excellent nucleophile. And so it's going to be, this sulfur is going to be cysteine is going to be one of our nucleophilic amino acids that we'll see. And so it isn't that strong of an acid, but it is still an acid. It can deprotonate, and it can deprotonate a lot easier than its kind of like hydroxyl version. So remember that cysteine is going to be basically the sulfhydryl version of serine. So serine looks exactly like this, except it has an OH here instead of an SH. So that O is going to be more electronegative. It's going to form stronger hydrogen bonds, but it's not going to deprotonate as easily. And so sulfur is going to be a better nucleophile for us, and it's going to deprotonate more easily. And remember that even though these are nonpolar, they can still form interactions. They can form those like transient dipoles. They can form those van der Waals interactions. They can form interactions that are going to be individually very, very weak, but collectively very, very strong, especially if you have a whole bunch of them clumped together after you've kicked out water. You get these, what we call like hydrophobic interactions are going to occur in here. So we have hydrophobic interactions, phobic interactions. And remember, those are just going to be like our van der Waals. So we're thinking about things like dispersion forces, just random transient separation of charge that leads to these very weak individual, but collectively strong bonds. So when we think about these nonpolar uncharged, we're going to be thinking about our van der Waals interactions, and we're going to be thinking about we're going to think about hydrophobic interactions. Not all of these are created equally. So we talked about how we could have aliphatic versus aromatic, and we'll get back to that in a second. But we can also have differences in their size and in branching. So if we take a look at valine versus leucine and isoleucine. With valine, so remember this valine, you can remember V, it has a kind of like V shape. If we were to go ahead and draw it with our stereochemistry, what we would see is that we have carbon's going to be sticking up out of this, and then we're going to have the carbon, the CH3 bond sticking up like this. And so basically you can think about it having this V structure. And so V for valine. But anyway, when we think about valine, and then we compare it to leucine and isoleucine, with leucine, basically we have one more carbon in between that V, that branching point, and the backbone. And with isoleucine, it has the same number of carbons and hydrogens as leucine, so it has the same like molecular weight, but the branch point is going to be closer to the backbone. So these are examples of what we call branched chain amino acids. And we'll see that these branched chain amino acids are going to have like similar breakdown pathways and stuff, but the consequence of having different branching point is going to have influence on what angles that their backbone can take. We'll get much, much more into this when we talk about peptides, when we talk about peptide bonds, when we talk about secondary structure. But when you link the amino acids together, we're going to have those side chains, those R groups sticking out. And then we're going to have these peptide bonds. So remember, peptide bond is just another name for an amide bond when it's occurring in the context of a protein or between amino acids. And it's going to have a partial double bond character. It's going to be stabilized by resonance. And remember that for a resonance stabilization, things need to be in a plane. So you're not going to be able to rotate around here. So you're going to get basically this chain of planes where what you're going to have is you're going to have specific points at which you can rotate, but just how far you can rotate is going to depend on things like the size of your side chain and whether that bulk is kind of concentrated near the backbone. So if we think about something like a valine or an isoleucine, it's going to have this bulk really close to the backbone, whereas the leucine, well, now we're kind of pushing that bulk further away. So even though it's bigger, it's kind of going to have a little more flexibility. And so we'll see how things like this can have an influence on the protein's shape and how it folds. Similarly, if we have other things that are really bulky near the backbone, so like tryptophan, those are going to be hard to turn around, but something like arginine, even though it's big, well, it's got its bulk kind of pushed away, and so it's going to be able to form more angles. We'll see how basically different amino acids, not only can different amino acids form kind of like different angles, so there's more likely places that they'll be, but amino acids typically form kind of conventional angles because this is going to allow them to maximize their hydrogen bond opportunities through the backbone interactions. And so we'll look much more at this and then how we can plot the angles on something called a Ramachandran plot. So we talked mostly about those like straight chain ones, those aliphatic ones, but we also have some of the nonpolar uncharged, as well as one of the polar uncharged ones are aromatic. So you can also call these like aryl. And basically these are going to have those resonance stabilized rings. Typically, when we think about these, we most often think about tryptophan, phenylalanine, and tyrosine, but technically histidine is aromatic as well, even if this is not dependent on its proteination state. So aromaticity is not dependent on its proteination state. So we'll talk in a minute about it as being one of the basic amino acids, but that is not going to be affecting its aromaticity because you still have a second, a lone pair that is going to be contributing to that resonance. What the aromatic amino acids, one of the things they have in common is that they're going to absorb UV light. And so we're going to see in the lab how we can use the absorbance of 280 nanometer light to measure the concentration of protein, and how this can be influenced one way or another by if you have more or less of these amino acids that are going to be the ones that are absorbing the light. Okay, so now let's talk about the polar uncharged. So when we talk about the polar uncharged, what we're talking about here, we're talking about our serine, threonine, asparagine, glutamine, and tyrosine. And what these polar uncharged ones have in common is they're going to have either a hydroxyl group. And we're also going to be talking about cysteine. So we're going to have a hydroxyl group, or we're going to have an amide group, or we're going to have a sulfhydryl group. So that'd be our thiol group. And what these all have in common is basically they're going to be able to do things like form hydrogen bonds, and they can act as nucleophiles. So when we talk about charged amino acids, lysine and histidine, when they're deprotonated, can act as nucleophiles as well. They have a lone pair. But whenever we have one of those hydroxyls or we have those cysteines, those lone pairs can form hydrogen bonds. And when these groups are deprotonated, especially, they're going to be strong nucleophiles. And we'll see how these amino acids can get like deprotonated in the context of an active site of an enzyme, and therefore make strong nucleophiles that can do things like attack peptide bonds and break them up. And so we'll see lots of examples of that. And remember that cysteine, although it's like pretty hydrophobic, we classify it as with the polar ones because it acts as a weak base. It can deprotonate. It can actually deprotonate more easily than a hydroxyl group. And it can therefore act as a strong nucleophile. And it's going to be able to form those hydrogen bonds because remember, you have those two lone pairs of electrons that can then serve as acceptors for hydrogens that are attached to something else electronegative. We'll also see that cysteine is going to be able to form crosslinks. So we can say cysteine like oxidizes to form these crosslinks because it's giving up its bond to a hydrogen for a bond to another sulfur. And so we're going to see these crosslinks of cysteine. And note that cysteine is not the only one that has sulfur. There's also methionine, but methionine is going to be one of our nonpolar uncharged amino acids. Methionine is going to have that sulfur as part of like a chain. And so it's not going to act as it's not going to act as like an acid or anything like this. But the cysteine can. But these are the two that have sulfur. So two have sulfur. And those would be our cysteine and our methionine. Okay, now let's talk about the charged ones. So we have the ones that are most of the time, they're negatively charged. These are going to be what we call our acidic amino acids. And we have the ones that are some of the time at least positively charged, and these are going to be our basic amino acids. And it's kind of confusing to think about in this term because what we're saying is this is going to be in neutral form when these are acidic, and then same with the basic, it's going to be basic in its neutral form. When it's in its ionized form, it's already acted as an acid or it's already acted as a base. So aspartate in its negatively charged state has already acted as an acid. It's given up a proton, and so now it's in its conjugate base form. So it can act as a base, but we call it acidic because in its neutral form, it acted as an acid. Similarly, with the basic amino acids, in their neutral form, they acted as bases, and they did what bases do, and they took a proton, so they become positively charged, and now they're in their conjugate acid form. They can act as an acid, not as a base. So it's kind of confusing, but you can get used to the names. We often represent this acidic ones, like for coloring a protein structure, the acidic ones are going to be red and the basic ones are going to be blue. You can remember this because the oxygens we often draw as like red and nitrogens we often draw as blue. And these acidic amino acids are going to have the acidic because they have those carboxylic acid groups, which are going to deprotonate, and these have these oxygens. And so we can think of them as red. And for these basic ones, well, the reason why they're basic is because they have some sort of amine group that is going to be able to take a proton. So it's able to act as a base, take that proton, and the nitrogen is going to be represented as blue. And so we get that we represent these basic amino acids as blue. So in terms of these three, arginine and lysine are almost always going to be protonated, whereas histidine, well, it's kind of more of a mix. And so remember to know what protonation state we're likely to find things in. We need to look to its pKa. And if we want to know exactly, we can use like the Henderson-Hasselbalch equation. But by thinking about for every one unit change in the pH, you're going to have 10-fold more of the acid or the base, depending which direction you go. So if we compare the pH to the pKa, if we're above the pKa, we're going to have less of it to be protonated. And if we're below the pKa, we're going to have more of it be protonated. In the case of a basic amino acid, it's the protonated state that's going to be charged. So if we're anywhere below the pKa in terms of our pH, we're going to be mostly positively charged. And when we think about lysine and arginine, they have pKa's of about 10.5 and 12.5. And so this is going to make it so that we're typically always at below the pKa. And so we're going to be several pH units below that pKa. So these are almost always going to be positively charged. But we will see examples how lysine can be deprotonated in active sites of enzymes and play really important roles as a nucleophile. So even though we think about lysine as if it was just hanging out by itself, it would be positively charged, we're going to see important examples where it does get deprotonated. In terms of histidine, its pKa is actually a little below like physiological, but it's only about 1 pH unit below. So we have about 10-fold more of the deprotonated, the neutral form, than the protonated form, but we're still going to have significant amounts of each of these. So histidine is this one that can kind of go back and forth. Predominantly, it's actually going to be in that deprotonated state, but it can easily be in either state, and we calcify it as one of those basic amino acids. In terms of our acidic amino acids, so these are the ones that, remember, in their neutral form, they're an acid, they have a carboxylic acid group. So we can refer to these as a glutamic acid and an aspartic acid when we're thinking about them in their acidic forms. But then these often, typically, they're going to be deprotonated to give you their carboxylate form. So their carboxylate form is going to be that conjugate base, remember, and it's going to be give the name ate. So we have glutamate or aspartate. And remember, you can think of aspartate or to remember that D. And then glutamate, it has basically, it's the next letter in the alphabet and it's one longer than the aspartate. And so you often see the terms like glutamate and glutamic acid used interchangeably, as well as aspartate and aspartic acid used interchangeably, because these molecules can interchange just by giving or taking a proton. But if we look at their pKa's are about 4. And so basically what this is telling us is that if we're at a pH of, say, 7, we're going to be several pH units above the pKa. So we're going to have like more than 1,000-fold more of it be in that deprotonated state. And so we're typically going to see it deprotonated. And remember that these are kind of just the carboxylic acid versions of the glutamine and asparagine, or you can think of asparagine and glutamine as being the amide versions of the aspartate and glutamate. Basically, they have the same kind of structure. They have the same number of carbons, but you're going to have an amide group at the end of an asparagine versus a carboxylic acid group at the end of an aspartate. And you're going to have an amide group at the end of glutamine versus an amide group at the end of glutamate. But for each of these, remember that the one that is longer is going to be the one that has the name further in the alphabet. So that would be our glutamate and our glutamine, which have 3 carbons after the alpha carbon, as opposed to your asparagine and aspartate, which only have those 2. Just a note while we're here is basically we can think about that as our alpha, and then we can refer to this as going to be our beta, and this is going to be our gamma, and et cetera, et cetera, et cetera. You can do naming like that for each of these carbons. But it's that the central one is this is going to be that chiral center. For some of the amino acids, you do have another chiral center further on. The one we care most about is going to be that alpha carbon. And then finally, coming back to our weirdos, we have glycine and we have proline. And so proline is going to be one of these. So it's nonpolar, but it's weird. And so it doesn't really act like most of the nonpolar ones. It doesn't act like the polar ones either. It just kind of like acts like itself. So we talked about how glycine had that really small side chain. It only had a hydrogen. You can think of it as not even having a side chain. It's not even chiral. Well, proline has a side chain that kind of like wraps in on itself. And when it wraps in on itself, it's going to make it so that it has very awkward angles. It's not going to be able to rotate much. And so in the case of this proline, and when it's bonded in part of a peptide bond, it's not even going to have a hydrogen in its backbone. So basically, this proline is going to be weird. It's not going to be able to form normal bonds. It's not going to be able to form normal angles. We're not going to find it in those common secondary structures. It's not going to be able to be found in those things like helices and strands. And so proline is going to be really weird. It's going to be good for like turns, as well as good for like stabilizing proteins. So we'll look at how like proteins can be stabilized by adding proline, stabilizing them in one conformation, and things like this. So remember, proline and glycine are weird. Glycine's weird because it has no side chain or it just has a hydrogen. It can form more angles than usual, so it makes things kind of floppy. And proline's going to make things really stiff and stuck in certain angles. And we'll look much more at these and do some interactive demonstrations with like Ramachandran plots and stuff. And remember that although those are kind of like the extremes, the rotation is also going to be influenced by the size of the side chain, as well as how close to the backbone that bulk is. So the closer to the backbone the bulk is, the less rotation you can have, the more restricted. So hopefully now you can now see how the properties of each of these amino acids are going to influence the final structure of the protein, and that final structure of the protein is going to influence the function of the protein. We'll look much more explicitly at how this happens in later posts, but all of it is going to be based off of those amino acids and the sequence of amino acids. So it's important to know these amino acids, know their structures, their names and abbreviations, know their stereochemistry, be able to classify them as polar or nonpolar, aliphatic or aromatic, and basic or acidic and neutral, and also like normal and weird. Also be able to kind of like tell something that's both closer or farther from its side chain and how that might influence things. And we'll get much more into that. You should also be able to identify the functional groups and recognize which amino acids have which functional groups without even having to draw them. So if you see that on paper, you should be able to go ahead and circle those functional groups, but you should also know without having to draw them. So we're talking about our things like our carboxylic acid groups, our amine groups, our amide groups. We're talking things like hydroxyl groups, sulfhydryl groups. You want to know which functional groups which amino acids have. You should also know which amino acids are nucleophilic. So remember, those are going to be your serine, your threonine, your tyrosine, your cysteine, and your lysine and your histidine. But know that those have to be deprotonated in order for them to have a lone pair available. And that the ones with the hydroxyl and the sulfhydryl groups are going to be much stronger nucleophiles once they've actually deprotonated. And that cysteine is more likely to do this.